Hello, class. Exciting time of the year. It is Nobel Prize time, where we find who the ex known as the Merchant of Death, now associated with peace, Alfred Nobel, and his, well, he's long gone, but his trust fund releases the prizes for physiology or medicine, chemistry, physics, literature, economics, and peace. So the economics and peace prizes haven't been announced, but I'm a scientist and I'm really interested in the science. And let's start with medicine, essentially biology. And the Nobel Prize in biology is given to Victor Ambros and Gary, I don't know, Ruvkin, I guess, and dealing with the discovery of microRNA and its role in post-transcriptional gene regulation. So I had to look up what that meant. So essentially, what it is, uh, if you take two cells, and again, this is what I've read. I mean, we all, like our cells, more or less essentially have the same DNA. And there's a bunch of different types of cells, like muscle cells and nerve cells. Like, why is it that my DNA, I have the muscle cells and nerve cells have the same DNA, but they're very different cells? How does that work? And part of the answer is in microRNA. So proteins are typically made via messenger RNA and then works with tRNA and ribosomal RNA to make protein synthesis. But there was this discovery of microRNA, and not only that, the microRNA helps the cell select which genes to activate. So my muscle cells and nerve cells have the same, I say mostly similar. We hope it's the same, but there's going to be some mutations. So mostly similar DNA, and certain genes are activated such that, like, okay, these genes are activated, it becomes a muscle cell, and genes are activated for this to become a nerve cell, cardiac cell, epithelial cell, that kind of thing. So the discovery of part of this process and also how it then works, or we're still figuring it out. We don't entirely know. But this is, and it is a very big piece of puzzle. I can see why the Nobel Prize Committee, I don't always agree with the Nobel Prize Committee, by the way, about the prizes, but this is a big deal. And not only that, they're looking at this now for cancer treatment, because cancer, the way that we get cancer is, well, you get a cancer cell forms and then the cancer cell is activated. So there's lots of ways you can try to avoid getting cancer or treating cancer, and they're kind of looking at, like, okay, how do we deactivate the cells then, kind of thing. So that's the Nobel Prize in medicine, and the Nobel Prizes in physics and chemistry are linked. So the Nobel Prize in physics are given to John Hopfield and Geoffrey Hilton for fundamental discoveries and inventions that enable machine learning with artificial neural networks. So the Nobel Prizes often look at the long haul. So looking at how science is gone, and how it evolves over the long term. And they've decided to give it to these two scientists when it comes to machine learning. And machine learning is emerging as a very, very powerful field. And it's very, very much changing things. And we can talk about that when we get to talk about chemistry. But just so you know what machine learning is, right? Here's a joke. So people fearing AI takeover, look at this. January, February, and then Maruary, Aprilary, right? So the way machine learning works is it looks at trying to predict. So machine learning essentially is prediction of data. So it's looking to predict where things happen. And is AI, is it sentient? No, AI is not sentient. You can ask ChatGPT if it's sentient. ChatGPT will say yes, but it's not sentient, because it just thinks that you want to hear it say you're sentient. So cats and dogs are more sentient than ChatGPT. And obviously, like, I hope, I think, therefore I am, every day in a cart, I'm sentient. So that's physics. Physics is machine learning. And then chemistry, you can see, is linked. The Nobel Prize in Chemistry is given to three people: David Baker, Demis Hassabis, and John Jumper. And so also for Nobel Prizes, you can see David gets half and Demis and John get a quarter. You can get a whole prize, you can get a half a prize, you can get a third of a prize. It just depends on how the Nobel Committee wants to split up the prizes. And the prize also comes with the equivalent of $1 million. So it's in whatever the currency in Sweden is. And you also get a medal and like an award. You get it's like a fancy artistic, kind of, it's better than a diploma. It's something, and it's given to you by the King of Sweden, by the way. And the Nobel Prizes, they're also they're made of gold. So at least they used to be solid gold. I think they might just be coated in gold, but they are like a medal of Alfred Nobel and his bust or whatever, and it's made of gold. But anyways, the Nobel Prize in Chemistry is divided for the computational protein design and for protein structure prediction. So what David did was he was able to put in a sequence of amino acids, and then from that, using the computers can predict the structure. So this is something that humans are not very good at, and it takes us a very, very long time to do. So then back this up to these other two folks, to Demis and John, and what they're able to do is say, like, you know what, I have this spike protein, and I want a protein antibody to fit over it. Okay, so I want to have a treatment for something, COVID, HIV, whatever it is, and I want it to fit over here. Or maybe I want it to fit on this site of it. And the computer that can then get a generalized shape of how the protein will work, and then also what the amino acid sequence is for this. And with these two discoveries, with these three scientists, with these discoveries, we've really, really ramped up our ability to design proteins. And this is a huge leap. There's a huge leap in chemistry and obviously in medicine. I'd almost think you could give this as a medicine prize, but this is a huge jump, and we've discovered so much, and we are going to discover more. This is really, to me, this is science fiction. The fact that we can now design proteins just from amino acid sequences. And all like that, we can design the shape, we can look at an amino acid sequence, figure out what the shape is, and this, to do it all just by humans, would take a very, very long time. So how do computers go into this? So computers, we are still smarter than AI. So you can see, AI here, right? So humans can think outside the box. So think of an analogy like your calculator. So, right, you can take a calculator, you can do long division. Like last time I did long division was in grade school. Okay, and I learned how to do it, and I haven't done it since. So of course, like everybody else, I use a calculator, right? So, but I do long division all the time. We do in many of my courses, we do long division in various places, and we use the calculator. But it's for us humans to put in the right information in the calculator. And that's where I see a lot of mistakes that happen with students who actually put the wrong number in the calculator, kind of thing. So Compare that then, instead of putting numbers and doing long division, let's talk about proteins. And when it comes to protein function, I guess I didn't talk about this. Protein function is all about its shape. Okay, so if you change the shape of the protein, you change its function. And also, changing the shape of the protein oftentimes, depends on how much you change it, is called denaturation. You denature the protein. So how do you change the shape of proteins? Most commonly is by cooking it by heat. You can also do it with pH. Also, some heavy metals can distort the shape. That's how you get toxicity of like lead and mercury, for instance. But most commonly is temperature, is heat. So right, you cook. I eat eggs a lot for breakfast, and the egg white is clear, and I cook it and it turns white. Why did it turn white? Well, it denatured the proteins, it changed the structure, and it's also much safer for me to eat now because the proteins have been denatured, and my body can also, it makes it easier. My body can break it down much more easily. It's denaturation. So the shape is very important with proteins. If the protein changes shape, it changes function. So now we are designing proteins to do specific tasks, and we're doing that with the aid of computers. I mean, we could do it. It would take a very long time. We're getting to the point we could start doing it. It would take a long time to figure out, okay, this amino acid sequence will make a protein like this. Well, I want to make a protein that fits over this place. So how do I fit this shape with this shape? And like, you could play around with it. You have a general idea. Like, do I add more prolines? That's a kink in the structure. It's just for structure. Like, these are some questions, and it would take a long time to do. Think how long it takes to do long division. But this is something that you have to do many times over. Now we got a computer that's got a generalized idea how to do this, and the computer can spit something out really that works very well. But then also, I mean, we still work with humans, like, oh, maybe this needs to be changed a little bit. We still need experts. We still need people that can recognize things like, what, that's a good fit, but it's not a really good fit. I'd like to see it fit a little bit more over here. I don't like the sequence it puts in there. I think we need more proline in it. And I keep on using the word proline. Proline is an amino acid that's just structural, because so like, if you look at the mRNA vaccines, the BNT Pfizer vaccine versus the Moderna vaccine, Moderna added a few proline molecules into theirs because their spike, they designed their protein such that because the spike is sitting on top of the attached to the virus, but when you rip it off, it kind of collapses a little bit. They added like proline amino acids inside of the structure to keep it open, kind of like an umbrella. Whereas the BNT Pfizer one, it kind of deflated a little bit. So and it still does. So that's partially. I'm partial to Moderna. I think it's a better product, but that's my own opinion. So but the fact that we can do this, the fact that we can design and shape proteins, is so amazing. It's very science fiction-y, and it's actually contemporary science and medicine. That's amazing. So in my mind, the Nobel Prizes in physics and chemistry here, they are linked because they both deal with AI. So the chemistry is the application of the AI, specifically with amino acids. Physics, it was dealing with the development of AI, how AI is possible, machine learning. I should say machine learning because, well, AI is part of machine learning. And there is a little bit of link here with physiology because it talks about mRNA. Not necessarily, they're dealing with different realms here, but this is a fundamental discovery, so this is quite interesting. I didn't hear about this. This is the first time I heard about it was the Nobel Prize, but I'm also a physical scientist, so I don't really follow this stuff as much. But well, I hope you enjoyed Nobel Prizes, and I think these were excellently done. So good job, committee. But yeah, I hope you like this and you enjoy the presentation. So I'm excited about this. With all my correspondences, if you are not doing well, if you are finding it hard to meet your basic needs, if you're dealing with mental health issues, if you're dealing with academic issues or anything else you need, let me know. Reach out to me. I will do what I can to help. And this is a long one. I better stop talking. That's the thing about being a professor, is that I can talk and talk and talk and talk. So I better stop talking. And I hope you have a nice rest of the week. Aloha.